Rituximab itself is a biosimilar. There's a very nice paper published in Nature Biotechnology in 2011, which showed they did serial analysis of Rituximab produced at various years and looked at this both biochemically and in various in vitro assays. And it's quite clear, from their analysis that Rituximab underwent a step change and became very different. I can't remember when it was, which year, 2010, I think. They changed the manufacturing, Roche changed the manufacturing, and Rituximab itself is a biosimilar, right? So there are very few, if you like, scientific reasons for thinking that we shouldn't be using biosimilars. Then both Celltrion and Sandoz have done extensive studies in patients. The Sandoz study is particularly worthwhile commenting on because there's so many numbers, there are large numbers of patients where they compare their biosimilar and Rituximab head-to-head in combination with CVP in patients with follicular lymphoma requiring treatment, and the results are entirely equivalent. And that's what we're looking for, equivalence. If they're equivalent in every parameter that we can assess, both in vitro and in the patients, there are very few reasons, in fact I can't think of any, for not using these antibodies instead of Rituximab. Some people might say that, well, we shouldn't be using them in diffuse large B-cell lymphoma because that's a curative regimen. But, equivalence, if equivalence applies in one disease, it applies in everything, in my view. And all the data to date indicate that certainly the Celltrion and the Sandoz biosimilar Rituximabs are equivalent. And therefore, there is no logical reason, no scientific reason, not to use these antibodies, even in the first-line curative setting. There are problems, of course. Because we use fast infusions now for Rituximab. There are no data that speak to that in terms of biosimilars, but that's a protocol that will develop with time. Obviously, it's not going to supplant subcutaneous Rituximab, right? Because that really is deep magic. How you produce an antibody at 100 milligrams per mil without it crystallizing out of solution, I have no idea. Right, and so I think that side of things is going to remain the same. But if for infusion or for IV Rituximab, biosimilars are going to take over, I think, because their price is, the Celltrion antibody will be about 50% of that of Rituximab. So a lot less. The price difference is a lot bigger than I thought it was going to be, and it'll be very difficult, I think, to argue against that. Anyway, it's going to be commissioned, so we won't have any argument about it. It will be a done deal. And it'll save the NHS about 150 million a year, they reckon, which is nice work if you can get it.